Good morning, good afternoon, good evening. Wherever this finds you, we want to welcome you to the 67th Emerging Growth Conference and day 2 of our 2-day Virtual Investor Conference. I'm Anna Berry. So just a few notes. Today we're running until 5 p.m. Eastern. And when we switch to the next company, you might see a black screen for a moment. Don't go anywhere. That's just us moving over to the next presenter. If you do experience downtime, refresh your browser. That usually puts everything back together again. Our platform does work best on Google Chrome. So if you're watching from an Apple device, you have to hit the play button to start the session. Now, all of our conferences, they're uploaded to our Emerging Growth Conference YouTube channel. So please subscribe, youtube.com/Emerging Growth Conference. Now, today, during each company's presentation, you can submit questions through the webcast module, and we will attempt to address as many of these at the end of the presentation. One last note. After today's event, you'll be redirected to the registration page for our next conference. So stay on or come back to reserve your spot early. Let's begin with Angle PLC. It trades on the OTCQX under the symbol ANPCY and on the AIM under the symbol AGL. It is a world-leading liquid biopsy company with innovative circulating tumor cell diagnostic solutions for use in research, drug development, and clinical oncology using a simple blood sample. So happy to bring on the founder, CEO, and executive director. We have Andrew Newland, and the CFO, Ian Griffiths, will join for the Q&A. Welcome, Andrew.
Thank you very much indeed, Anna. And I'm very pleased to have the opportunity to present to you this morning. Thank you very much for the invitation. So I'm going to describe a new approach to cancer diagnosis and treatment. Now, as Anna mentioned, we're a UK-listed company on the London Stock Exchange, and we also have a listing on the OTCQX ANPCY. Despite being a UK company, we have very strong interests in the United States. And in fact, our two largest shareholders on the current register are, in fact, United States institutions. We've had a very long involvement in the US. In fact, the original technology for Otcqx, which is the name of our system, was in fact the first patent filed in Philadelphia. So we're very much associated with the United States. And what you're looking at there in this very first slide is a side view of the Otcqx instrument. And our system comprises an instrument and a one-time use consumable. And what you see in the middle of that image is a tube of blood. So that's a standard tube of blood drawn from a patient arm in the normal way. And in that blood, if the patient has cancer, there will likely be a small number of cancer cells, maybe 10 cancer cells in a tube of 7.5 milliliters. So roughly speaking, there might be one cancer cell in 1,000 million blood cells. So it's a very tough job to get such a rare number of cells out of the blood. But if you can retrieve them, which we've proven out with our Otcqx system is possible on a reliable and reproducible basis, then what you can do is via a simple blood test, you can obtain information on the cancer development. So how is it progressing in the patient? Now, it's kind of shocking, but if you think about the way that cancer patients are treated at the moment, standard of care involves a tissue biopsy. So that means cutting out some of the cancer for the purposes of analysis. And the reason that's important is in order to distinguish one patient's cancer from another. So I often use the example of breast cancer, but this system works without modification for all solid tumor cancer types that it's been used with. But in breast cancer, for example, you imagine that 3 women with breast cancer have the same disease. Well, actually, in reality, that's not the case. They have a disease in the same part of their body, but it's not the same disease. So they will respond differently to different treatments, and the doctors have to make quite complex decisions as to which standard of care pathway the patient should go onto. That's all very well and good, and that works fine. But the challenge is not only does cancer vary between patients, but it's also dynamic, and it is in a state of constant change. So cancer will present in a particular way, but sometime later, maybe a few months later, it may be fundamentally quite different. And that, in fact, is what's shocking, is that to all intents and purposes, the doctors have to work off the original tissue biopsy that they got. They can't get new information to guide treatment because those patients were likely to have had surgery to cut out the cancer. And if it's been cut out, you obviously cannot go back and cut out some more for a tissue biopsy, unless, of course, it's spread to a secondary site, which I will mention shortly. So what we can do with a blood test is recover some cancer cells for analysis. And the reason the cancer cells are there, of course, is that that's how cancer spreads. And if you look at on patients whose cancer has unfortunately progressed and they end up dying from cancer, they don't die from the primary. So they don't die from the original location. They die when it's spread to metastatic sites. So in breast cancer, it can spread to the bones or the brain or the lungs or the liver. Those are 4 common metastatic sites for breast cancer patients. And it gets there via the blood circulation system. So the cancer cells that are in the blood that we can recover with our system, which are known as circulating tumour cells, are actually the seeds for the metastatic spreading of the disease, and therefore they're critically important to provide information. So in terms of the status of our company, we have put a great deal of effort into getting a US FDA product clearance. In fact, we took over 6 years of work, 16,000 samples, in order to secure an FDA product clearance. And we're the first and only company in the world to have an FDA product clearance for recovering cancer cells from patient blood for subsequent analysis. And off the back of that, we've actually been very successful in building out a distributor network and starting to grow product sales. So we're selling this machine and the consumables associated with it worldwide. We've also had, and I will describe it to you, some breakthrough data showing the advantages of doing combined DNA analysis of the sample type that can be accessed with our Otcqx system. And in addition to all of that, we've built out a services business providing services to pharmaceutical customers who have got cancer drug trials. And I'll explain how that works as well. In 2024, we're looking to building this position very substantially. So we're looking for wider use in pharmaceutical clinical trials. We're seeking to get clinical tests being offered for patient management from Angle's own clinical laboratory later this year. And we've actually got 2 important cancer studies going on to do with the detection and assessment of cancer in prostate and ovarian cancers. Now, the liquid biopsy market, it's called liquid biopsy because it contrasts with tissue biopsy. So tissue is where you cut it out, and liquid biopsy is where you get it from a liquid, which is in this case blood. So there have been independent analysts who have assessed that the United States market for liquid biopsy is actually in excess of $100 billion per annum. So it's a very large market. And of course, this is applicable not just in the United States, but worldwide. So probably 3 times that in the wider marketplace. So in terms of our FDA product clearance, the clearance is specifically in relation to metastatic breast cancer, so cancer that's spread. Nevertheless, this system has been shown to work without modification in 24 different types of cancer. And we believe that recovering intact living cancer cells from patient blood is the best possible sample for repeat sampling of what's happening with the cancer to drive treatment decisions to how best to improve the patient's outcome. So what you see there is the instrument. It's about the same size as an inkjet printer that you might have at home. And then the consumable, which is the same size as a microscope slide. It's a very smart, but nevertheless relatively cheap to make, proprietary microfluidic cassette, and the blood flows inside that. So in terms of what's happening in the near term, we're expecting to, which is why we believe it's important for us to spread information to prospective investors and provide updates at this time. We've got a number of important commercial milestones coming through. We just secured our first large pharmaceutical customer, so we've got several other smaller ones, but this is a $4 billion revenue customer, and Eisai, which is a very large Japanese customer. We've got other contract discussions going with other large pharmaceutical companies. So we're expecting to see our services business begin to take off. We're also building out our product revenue. So we've now established our distributor base, and we've also launched a Portrait Plus product kit, which I'll explain as well. We've got breakthrough molecular results, which we've achieved with the Illumina next generation sequencing platform. But the news to come is that we're working on other molecular platforms, including the Thermo Fisher next generation sequencing platform. And in addition to that, we're running ovarian and prostate cancer studies where we've already processed samples from patients with the Parsortix. And these are held in a freezer pending analysis with a third-party molecular platform. We're working to determine which is the most appropriate platform to do that, and we anticipate getting those study results later in this year. And we're also moving to get our Angle clinical lab in a position so that we can offer tests for patients. So there's a lot to play for in the current year's activity. So just to sort of recap on the flaw in standard of care that we're addressing, the National Cancer Guidelines in the US recommend a biopsy, a tissue biopsy, for all metastatic breast cancer patients. So what that means is if the cancer has spread to one of the secondary organs, then the patient should have surgery, which involves general anesthetic, in order to cut out some of the cancer. So as an example, if the cancer has spread from the breast to the liver, then the patient will have general anesthetic and surgery to cut out some of their liver. That's not to improve their outcome as such, it's in order to recover cancer cells for analysis, because the guidelines understand that the cancer's most likely changed significantly from where it was on the primary tissue biopsy. So they need more information. But despite this being in the guidelines, over half of all of the metastatic breast cancer patients do not have a successful tissue biopsy at this point because they're either too sick for the surgery, which is often the case, or the tumor's inaccessible, or there's not enough tissue to cut out. So the lady on the right-hand side of this slide, Dr. Julie Lang, who's the chief of breast surgery at Cleveland Clinic, she has done the first ever comparison of RNA gene expression of cancer cells coming from a blood sample with matched paired samples from biopsy of metastatic tissue samples in metastatic breast cancer. And what she basically found was she could get the same RNA gene expression information from a blood test using our Parsortix system as was possible to get from this invasive surgery. So ideally, patients wouldn't just have one time point additional, they would have a routine test every 3 months or every 6 months if they could get these cancer cells out. And that's what our system has been successful in achieving, is enabling the recovery of the cancer cells. And the way it does it, this is a little diagram, a schematic of our Parsortix cassette. And the blood flows inside that, so it goes down an inlet channel, but the channel's closed at the end. So it has to go either left or right in order to reach the outlet. And that takes it up a cross-section with a series of steps. So this is a proprietary, fully patented technology, so no one else can copy this approach. And the red and white blood cells can flow up the staircase and then through the critical gap. And they then make it into the outlet channel and flow away. So the cancer cells are larger and less compressible than the blood cells, and they don't make it through the critical gap. Now, our machine automates this whole process in such a way that we can basically control the process. So when the blood has flown through, then we can actually, the machine will automatically flow a wash behind that blood in order to wash away any remnants of blood cells. Then the machine will allow the reverse flow out of the system, and that will flow back out the cancer cells. And you get the cancer cells out in prime condition. They're undamaged and they are available for analysis in that format. So on the next slide, the next slide, you see a picture of the instrument, and the instrument's automated all of this process. It's a very simple product to use. It's broadly speaking plug and play. You take a standard tube of blood, remove the top of the tube, attach it in the center of the instrument here, hit go, and the machine will advance the blood for you through one of these Otcqx cassettes, which is shown, it's held in the clamp here. The machine automates all that. So we've actually got a single technician is capable of running 20 of these instruments simultaneously. So it's possible to multiplex multiple samples at the same time. And because the samples come out completely undamaged, they haven't got anything attached to them, we haven't used any antibodies at this point to recover them, and they come out alive, undamaged, and in the same form as they were in the patient, in the perfect form for subsequent analysis. So we're taking advantage of this system by setting up distributors, and these distributors are very well remunerated. They get 30% of our gross revenues for any sales that they make, and this means that we're now just been able to start extending our reach and making sales in far-off countries such as China, India, Australia, etc., building on a current base in Europe and the United States. So that's really encouraging, and the distributors have been selected based on their existing oncology customer base, so customers who are already interested in this field, and they're beginning. We've trained now over 75 distributor staff members in order to do this kind of work. Now, we've also got a services business. The last one was the products business. This is the services business. And this is an accelerator and demonstrator, meaning that we're able to demonstrate new methods of what you can do with the cancer cells to have an impact, and also an accelerator in terms of getting quicker to market and generating revenues faster. And the primary customer base for the services industry are the pharmaceutical companies. And the reason the pharmaceutical companies are interested in this is because it can give them longitudinal information on patients in their trials. That means that they can assess the patient condition before they, as a baseline, before they come into a trial. Then they can assess it again during the trial and assess it afterwards. And the baseline offer that we've got is to assess what type of cancer cells are present in the patient blood, how many of them are there of each type, and then we can look at how that changes over time. In addition to that, we can look on the cancer cells for particular drug targets. So we have an assay development capability where the pharmaceutical company will tell us what their cancer drug trial is looking for, how does their drug work, and then we can look for the presence or absence of the protein of action for their drug. And in terms of the customer base that we've got, this is some examples. The first one I already mentioned, the Eisai, the Japanese pharmaceutical company, and we've been employed by them at this stage. It's a pilot project involving 50 patients, 2 time points, for which we're being paid $250,000. So we're getting $2,500 for each tube of blood that's processed, which is about half of what will be the ongoing price to them going forward. That pilot study, if successful, will lead into a phase 2 trial, which will probably be $2 or $3 million of revenue opportunity, and then into a phase 3, which might be $5 million. The end game, of course, is if the company wants to have a companion diagnostic to determine which patients should be given its drug, then it could be hundreds of millions of dollars of future revenues for Angle just from this one project. The project itself is a HER2 targeting antibody drug conjugate. Most sort of traditional cancer drugs like chemotherapy are very blunt drugs, and they generally attack fast-growing cells. The problem is that the body has many fast-growing cells, including, for example, hair follicles. Hence, patients suffer bad side effects from these drugs as healthy cells get killed. Now, the idea of an antibody drug conjugate is that the drug is carried in a drug delivery system that will bind onto cells that express HER2. So in this case, the targeted antibody is HER2. HER2 is a protein which can be expressed on breast cancer patients. It's expressed on about 25% of patients when they're diagnosed HER2 positive, and probably another 35% are HER2 low expressors. They're both targets for this drug. But 30 to 40% will not have HER2 expression of any sort whatsoever. Now, the critical thing is the HER2 status changes. 38% of patients' HER2 status will change over time. And what that will mean is that Eisai really does need to know what is the HER2 status of the patient in order to make sure that their drug is appropriately prescribed. And that's what we can look for. So we can look for the expression of HER2 on the cancer cells that come from blood, and we can then rank the patient as to whether they're HER2 positive, HER2 low, or HER2 negative. We're also working with a very strong company in the UK called Otcqx that's developing DNA damage response pathways, and we've developed an assay for them in proteins. And there's various other things. I'll keep moving in the interests of time. We've developed an antibody staining kit, which is designed to help our customer base use the antibodies that we've optimized for assessing the cancer cells. So this is if you like, a recipe that they can use to stain cells and look at them under a microscope. So the idea is that the customers who buy the Otcqx system can now mimic the work that we're doing and assess the status of these cells. We're expanding that now. We're developing a HER2 assay kit. So over time, we will be able to sell kits to laboratories all over the world so that they can repeat the HER2 studies that I was just describing. Now, moving into DNA, just one more bit of science jargon. Most of the companies, in fact, just about every company in the liquid biopsy space is focused on ctDNA, and there are multiple multi-billion market cap companies in the US who focus on ctDNA. So those are fragments of dead cancer cells. That's because our bodies are made up of millions and billions of cells. They go through a life cycle. At some point, some of them are dying and go into the blood, and then they get excreted with a half-life of about an hour. If a patient has cancer, they may have some cancer cells dying as well, and that's what's called circulating tumour DNA, or ctDNA for short. So all the industry is looking at these fragments of dead cells because they're easy to get hold of. But of course, it would be far better if you could get the actual living cells, which are spreading the disease. So we've just published data where we've done a direct comparison of ctDNA and ctcDNA, so dead and living, from the same tube of blood from the same patient, sequenced with exactly the same sequencing technology. And the hypothesis is we might find some information in the living cancer cells, which is not available in the dead fragments of cells. And the truth is, that's exactly what happened. So we found that 70% of breast cancer patients had actionable DNA variants, so mutations, in their blood from the living cancer cells, which were not evident in the circulating tumor DNA dead fragments. So what that means is this patient has got clonal evolution, new DNA mutations coming through in their cancer, which are spreading the disease in the metastatic circulating tumor cells, which they cannot see from the tissue biopsy on presentation, and they cannot see in the ctDNA. So this is clonal evolution, and it is how the cancer is evading the immune system and evading the therapies. And we believe that this has tremendous importance for future personalized medicine for cancer patients, to know how their cancer is changing and how to address it. And indeed, what was incredibly exciting is we found that the active DNA variants, these mutations that were in the living but not in the dead, including some very well-known mutations like PIK3CA, EGFR, ESR1, mTOR, and ERV2. Now, these mutations are not unheard of. They're very well understood in tissue. And in fact, there are blockbuster billion-dollar drugs that have been promoted by the companies listed here: Novartis, AstraZeneca, Menarini, etc. Roche, Genentech, Puma, and Boehringer Ingelheim, just to name the ones that are in this list alone. So these pharmaceutical companies have got the potential to extend the offer of their drugs to more patients, because patients do actually have actionable mutations that their drug can hit, which at the moment the doctors don't know about. So it can be good for the expansion of the pharmaceutical company's sales, and that's a target for us as customers. But of course, it's critically important for patients as well, because at the moment, at a certain point, patients get into a palliative stage where doctors say, well, we can't do anything more for you. But if there was a blood test that found one of these actionable variants, well, possibly they could do some more for the patients. So we're driven very much by making a difference to benefit patients, and also, of course, to focus expenditure in healthcare to the right areas. Now, in our field, the currency really is peer-reviewed journals, publications by leading cancer centres. So what we've done is we've had our system in the hands of some of the world's leading cancer research centres and had them work with it. And we now got the benefit of 41 independent cancer centres who have published 92 peer-reviewed publications on our technology, all of which were positive, and almost every single one of these is available on our website if you want to dig into them. They are a pretty tough technical read, but nevertheless, they're very important. And they demonstrate benefit potential with 24 different solid cancer types. So there really is a wide open opportunity now to transform the way that cancer is diagnosed and treated going forward. And as I said, we're now at a stage where we're building out our services revenues and our product revenues. We've got some more breakthrough molecular results on the way, Thermo Fisher and some other platforms. Plus, we've got some very important clinical studies. We previously had a best-in-class ovarian cancer detection study of 95% area under the curve, which means basically very, very high accuracy of detecting ovarian cancer. So we've got those studies coming through later this year, and we will soon be offering patient tests from our own clinical laboratories. So that's the overview story. I hope you've enjoyed hearing some of this information. There's a lot more on our website, and we're happy to take email questions. But now, Anna, if you can take over and give us the Q&A, and Ian, if you'd like to join, please, our CFO.
Wonderful, Andrew. Thank you so much. We've got lots of questions for you. Let's see how many we can fit in. I'm going to try to combine a question here. Mark Sanchez asks, why do you need FDA clearance if the cells are recovered after the blood is taken? Is the method by which the blood is taken different from the norm? So how does the FDA clearance impact users?
So the FDA clearance is that we are able to do what's said on the tin. And what it says on the tin is we can recover cancer cells from the patient blood sample. So the actual drawing of the blood is absolutely standard. And cancer patients routinely have blood drawn for many different uses. This is one additional tube, that's all, from the same blood draw. And then the FDA clearance is basically demonstrating that we can successfully and reproducibly recover the cancer cells in the way that we described, and they can be taken out of our system and used for any form of downstream analysis to look at proteins, DNA, RNA, or morphological analysis.
Kim Beck wants you to explain in more details. Once the cells are recovered, what is the next step? Is what comes next as far as diagnostics standard procedure?
Yes, so what we've done is we've taken sort of standard procedures for tissue biopsy analysis of cancer cells, and then we've tried not to have to modify them at all, actually. So where possible, we're just using that same standard approach. Some aspects are slightly different because we've got a small number of cells, and we're also got them isolated individually rather than in an FFPE condition. But broadly speaking, it's the same approach, and you can look under a microscope. You can see these cells in the same way you can on a tissue. You can also do sequencing and PCR and other downstream molecular techniques.
And I'd add to that, that's a deliberate approach to work with third-party systems, because obviously they've already got an established installed base, credibility on the analysis. What we're doing is solving the repeat sample type and producing that better sample, but we feed their systems, and of course that makes the instrument organizations very interested as well, because we're able to help them add content to what they're doing and drive their sales.
Another science question from Jared Harrison. He says it's an ingenious approach, but will the cancer cells stop the following red and white cells since they are so large? And after the procedure, are only the cancer cells remaining? If you can clarify.
Yes, absolutely. I can clarify that. So the microfluidic technology that we showed there enables the red and white blood cells and the plasma liquid to continue flowing very, very easily right the way through the device. It's only the larger, less compressible cancer cells that are held onto. in terms of the output from Parsortix, there will be some residual white blood cells. There's be however many cancer cells that are present in the blood, 10 or 50, for example, or 1. Then there'll be some background white blood cells, maybe 2,000 to 3,000, depending on the blood tube used.
Yeah, just to elaborate on that one a little bit more, there is a video on our website of a real blood sample being run. If you look at that, you'll see that the step design, it's one really, really long step. So it captures the cancer cells, all the other cells flow either side of it. So do look at the website for the video.
Thank you. Al Bennett has a question about the revenue. Can you explain the different verticals and define them? What does the procedure cost exactly?
Ian, do you want to do that?
Yeah, so as Andrew mentioned, we've got both a product side and a service side, and they have different pricing associated with them and different tests. So if we take the product side, what we're trying to do there is sell into leading research use organizations, the cancer centers we've mentioned, into labs that then can use Parsortix to actually develop their own lab developed test, which has the lab developed test pricing. So just to give you a flavor on the product side, the standard pricing for the Parsortix research use is about $70,000 for an instrument. Cost of goods are about $15,000. For the clinical use, which we mentioned in relation to breast cancer, actually it's a bit higher. It's about $80,000 for an instrument. Still cost of goods of about $15,000. But it's a razor blade model. So if you look at the cassettes, the commercial use price for that is $300 for a cassette, and our cost of goods to manufacture that is about $15. We used a specialist nano manufacturer based in Germany, if you then add on the fact that we're using a distributor network, I think Andrew mentioned that it's 30% to the distributor, so we've still got a very healthy margin on that 300. If it drops down to 200, we're still making about 93% gross margin on that. So that's on the cassette side of things. So that's about us driving that side of the product base. The antibody kit Andrew mentioned as well, that's fairly cost effective. This is about trying to make it easy for people to use. So that's more straightforward. In terms of the service, it's variable pricing because it will depend what is the volume of tests that are required, what is the downstream analysis that's required, and is it a multiple types of analysis as well. So for example, the biodetect Andrew mentioned, if we're doing next generation sequencing on 2 different analytes, the CTCs and the ctDNA, our cost base is a lot higher, and hence the selling price is a lot higher. So, but if we take a range of the different types of downstream analysis, our selling price will be anywhere between $1,000 to $5,000 per test, dependent on the downstream analysis requirement that's there. And the gross margins will, again, depending on what we're doing on there, will be anywhere between 50 to sort of 85, 90%. And in terms of all the tests, as we've said, so far it's worked with every different cancer we've tried. We're a small company, so we can't focus on all of the cancers. Our leading cancers are the breast cancer, which is where we've got the FDA clearance, the ovarian cancer, where we've got the best-in-class results for the area under the curve, the 94.5% accuracy, prostate cancer, where we've had some really, really good results, and we've got a partnership developing with Solaris in the US, and also in lung cancer, which is a particularly difficult cancer, where we've got very good results as well. Sorry, wow. It's not an easy one to answer because there's so many. There's given they're both the sort of the horizontal and the vertical with the product and the service, and then if you look at the verticals for all the different tests, we can pretty much do anything.
Wonderful. Well, I think that answered the questions from our viewers. We have a few more, but we are out of time. Do you have a quick closing remarks for our viewers today, gentlemen?
I'd just like to thank you for your attention. We've got a very big focus on building this out in the United States. So it is important for us to get some retail and other high net worth investment in from the US. So thank you very much for your interest in what we're doing.
All right. Thank you. Important work you guys are doing, and we would love to have you on in the future with some updates. Thanks for joining us today.
Thank you.
Cheers. Bye.
Everyone stay with us. We'll be right back with Clear Day.